I think the next talk is also on remote. Is from Lei Zhao from the ICN2. The title of his presentation is Printed Electrodes for Bacterial Detection based on a smartphone platform. Thank you very much. The floor is yours.
Good afternoon. Now I will talk about printed electrode for bacterial detection based on a smartphone system. I will introduce the work in the following 4 parts. First, some background. Bacterial contaminations is one of most serious problems in environmental protection, food safety, and health care. According to a report, when the time reaches 2050, bacterial infections cause death will be the most serious risk. So this severe situation calls for early diagnostic system tools to reduce the healthy problem. For the bacterial detection, the commonly used method including PCR, ELISA, culture, and counting methods. Despite its university, this method has its restrict. On the other hand, electrochemical sensors is a good alternative because it can realize rapid, inexpensive, sensitive detection of analytes. However, for the electrochemical measurement, it's usually involving bulky equipment such as CHI and outlab. So in order to produce easy to use, cheap sensing platform, we came up with a fully printed system with a mobile phone readout. So basically, we produced reduced graphene oxide working and counter electrodes using inkjet printing method to produce the silver chloride reference. Then with a small portable PCR platform, we can connect it with a mobile phone by Bluetooth. Then for the sensor fabrication, it's a typical sandwich structure. First, we immobilize capture antibody, then it will recognize the bacteria, which is we used E. coli as a sample example. Then after this, we put a HRP labeled antibody. So this HRP will catalyze the reaction between hydrogen peroxide and TMB. After applying a potential, we can record the current signal. Then this current signal can relate it with the bacterial concentration. So the advantage of this system is we are not only use graphene as functional materials, also as a signal collector. The fully printed system enables the quick mass scale electrode production, and also the portable smartphone-based system can be a permission for point-of-care detection. So how we make the electrode? This work was developed based on previous work done in our group. Basically, we first form a graphene oxide film on the substrate, then using a laser to reduce it. After applying a pressure, we can selectively transfer the RGB electrode onto another surface. Following this, we use inkjet printing technology to produce the connections. Then after the passivation, we chlorinate the silver to form a pseudo-reference electrode. We first characterize the morphology of the materials. As you can see here, from the SEM picture, the graphene oxide film is quite homogeneous with a thickness around 10 micrometer. Then after applying the laser, a foam-like structure was formed with a thickness around 15 micrometer. After we transfer it to a different substrate, the film still keep a porous structure with a much less thickness. Then this trend was also confirmed by the height profile meter. Then we test the materials with a 4-probe system, get sheet resistance around 19 ohm per square, which means the AgCl has a good conductivity. Then the reduction was also confirmed by Raman. As you can see here, for the graphene oxide film, there's the typical D band and G band of graphene-based materials. After applying of the laser, the G band increased dramatically, and also the 2D band starts showing up. With the XPS analysis, we found that after the applying of the laser, the oxygen component was reduced a lot. This proves the graphene oxide was successfully reduced. Then we also characterize the inkjet-printed connections and the reference electrodes. As you can see from the resistance change here, that the connections the connection path has a good conductivity, and the SEM picture shows the clear morphology change after the chlorinization of silver. We also compared the reference with the standard commercial silver chloride reference electrode. As it shows in this curve, that within half an hour, there's nearly no potential change compared to the standard reference electrode. Then we checked the electrodes with electrochemical way. From the CV curve, you can see that with the presence of ferrocyanide, it shows a typical redox peaks. Then the electrode even can respond to a super low concentration of the redox probe, which is 5 micromolar. And the change was also confirmed by impedance measurements. The small standard deviation shows the, sorry? Okay, I thought I have a connection problem. Then the small standard deviation shows the good conductivity, reproducibility of the electrode. So after the characterization of the electrode, we use our system, did a test for the E. coli detection. As you can see here, there's a clear difference when E. coli was present and absent. So in conclusion, we combine laser-scribed laser printing method and inkjet printing, realize a quick chip fabrication of the electrode, and the smartphone-based system shows great potential for the point-of-care bacterial detection. So the next step will be optimize the conditions to reach the sensitive detection of E. coli. So finally, I would thanks all the group member from Upper Molecules group, and thanks for the funding from MicroPredict. Thank you so much.
Thank you very much. It's time now for questions.
Yeah, thank you very much for the interesting talk. I was just wondering, I like a lot, yeah, the process that you were describing with how you reduce the graphene with the laser, the CO2.
Yes.
So I don't know how does it compares with the, with the other chemical reduction of graphene, because it's very fast, no? Yeah, and I don't know how is if you can tell something about if comparing these two different methodologies, using the laser and using the typical chemical reducination.
Okay, so actually for the laser reduced method, basically it's a thermal method. When the laser was applied on the graphene oxide, it will generate instant high temperature. So then using some, then reduce the graphene oxide. So I think the main advantage is when you using the traditional method to reduce the graphene oxide, you reduce the materials and you need other method to deposit it on a standard like cold electrode or copper plus electrode. But in the laser reduced way, since it works basically as a printer method, so you can design whatever like patterns using a digital to pattern method. So you don't need like mask or other deposition tools. I'm not sure if I answer your question clear.
Basically, was also, I wanted to know the fundamental behind, yeah, the CO2 laser, how it was reducing, that you were saying that is a thermal, no? That is
Yes, yes.
Thank you.
Thank you.
I have another question. I mean, it's a bit late, but just let me, in the introduction of your presentation, you have shown that it seems that the intoxication from bacteria is increasing a lot, and also that the death rate for bacteria is going to overcome cancer. Is that true?
Sorry, I couldn't hear you.
In your introduction, you have some figures about the impact of bacteria on health. And if my question is, what is happening?
You mean here?
Yes. Why the number of deaths is increasing, and why you think that in few years bacteria are gonna be more dangerous than cancer?
So these figures come from a review paper. So it's describe the bacterial infections from during a specific period, and also they did a prediction of the serious problem, and it will be overpass the cancer. Honestly, I don't know exactly what's the reasons they did this prediction.
Quite scary, no?
Yeah.
Well, let's thank the speaker, and let's thank all the speakers of the session again, please.
Thank you.
Now the session is closed.